By the way, I like Haley's icon on the Zoom call. The Lego scientist.
Thanks, Maria.
I felt that it deserved a shout out. Oop. We are also streaming live on YouTube, so everyone, you know, just in case you were curious. GT, neither have gotten back to me, so.
Okay, thanks, Maria. We'll get going here. Want to call the emergency meeting of the ICOC and Application Review Subcommittee for April 24th to order. Hope everybody continues to be staying safe and healthy in this unsettling time. Just to recap, this is the third in the series of emergency meetings that we've had dealing with the COVID crisis. The first was March 27th, where we determined we wanted to do a funding round. The second on April 10th, where we discussed the topic of convalescent plasma as a potential vital research opportunity. And then third, oh, hi, David. That's an excellent looking friend you have in your lap there.
And Zelda is with Lauren. So I think it was bring your dog to the board meeting day, which I love. I encourage.
Excellent. Well, having said that, why don't we segue? Maria, will you please call the roll?
Anne-Marie Dulege.
Yes.
Isabel Duron.
Here.
David Higgins.
Here.
Steve Julesgaard.
Here.
Dave Martin. Lauren Miller.
Here.
Adriana Padilla.
Here.
Joe Panetta.
Here.
Francisco Prieto. Robert Quint.
Here.
Al Rolette.
Here.
Jeff Sheehy.
Here.
Oz Stewart.
Here.
Jonathan Thomas.
Here.
Art Torres.
Here.
Diane Winokur. Really quickly, David Martin, I could see that you're here, but we couldn't hear you, but I see you're connecting to audio. That was just an FYI. So hopefully we'll hear you soon. Okay, JT.
Thank you, Maria. So at this, our third meeting, we're going to be taking up the first round of projects. Before I turn the meeting over to Jeff to run the Application Review Subcommittee programmatic review, we just have a housekeeping item we've had at each of the last two, which is an action item which reads, consideration of existence of emergency situation. Emergency situation means any of the following is determined by a majority of the members of the Independent Citizens Oversight Committee. A, work stoppage or other activity that severely impairs public health or safety or both. And B, crippling disaster that severely impairs public health or safety or both. Do I hear a motion to adopt this item?
So moved.
Who was that?
Thank you. Yeah.
Okay.
I'll second it. I'll second it, San Diego.
Okay, David. Thank
Thanks,
you.
David. Is there any discussion by members of the board on this topic? Hearing none, any comments by members of the public on this topic? Hearing none, Maria, will you please call the roll?
Anne-Marie Dulege.
Yes, I agree.
Isabel Duron.
Yes.
David Higgins,
Yes.
Steve Julesgaard,
Yes.
Dave Martin, Lauren Miller,
Yes.
Adriana Padilla,
Yes.
Joe Panetta. Joe? Are you on?
Yes.
Oh, thank you. Francisco Prieto, Robert Quint,
Yes.
Al Rolette,
Yes.
Jeff Sheehy,
Yes.
Oz Stewart,
Yes.
Jonathan Thomas,
Yes.
Art Torres.
Aye.
Motion carries.
Thank you, Maria. We'll proceed to item 4, turn the meeting over to Mr. Sheehy.
Thank you, Chairman Thomas. So I think Dr. Sembrano, are you the one who's going to take us through the slides and the presentation on the review?
Yes, that's me. Thank you.
Great. I'm turning it over to you so we can see the results of the review.
Okay. Doug, are you going to put up the presentation or should I?
I'll set it up right now.
Thank you. Okay, just go ahead to the next slide. Okay, so good morning, everyone. We are meeting again regarding the COVID-19 program, and now we're bringing for your consideration recommendations of the grants working group for applications that came in response to this program. And so just as a reminder, we launched this a month ago, and the goal was to support projects that span from discovery through clinical stage, and we are utilizing our established partnering opportunities within those programs, the DISC-2, the TRAN-1, CLIN-1, and CLIN-2, to facilitate the application review and funding process. And as you'll recall, the board set aside $5 million in support of the program. Next slide, please. Next slide, please. Doug?
I don't know if his screen might have been.
It's moving forward. Do you not?
It's
You don't
not.
see it.
No.
Okay. Okay, well, anyway, on the next slide, there's a table that basically summarizes the overall program. And just again, this is a reminder of the 4 different types of programs that one can apply for. At the very top, starting with a clinical trial or clinical studies that provide an amount of up to $750,000 with an award duration of 24 months, all the way down to discovery, which provides up to $150,000 for up to 12 months. But for all of these, next slide, please, we're also having a requirement because of the urgency of this program that all projects must propose to achieve a clear deliverable within 6 months. And so this is really a bit of a higher bar than our typical programs in that we really are looking for projects that are ready to go and are ready to deliver something quickly. So the 6-month goal for each of these, for example, with the CLIN-2, is we expect that the projects will initiate enrollment and begin data collection. For a CLIN-1, that their IND filing will occur at that time point. For TRAN-1, that their pre-IND or equivalent interaction with the FDA occurs. And for the discovery ones, that they have data with a viable candidate that could progress quickly to the clinic. Next slide, please. And as you'll recall, 2 weeks ago, the board also allowed an increase to the program scope to allow consideration of investigational studies with convalescent plasma. And, you know, we normally don't fund projects that are not stem cell based. So because this is not a stem cell based type of project, we have to consider whether this represents a vital research opportunity in order to fund it. So although we're allowing them, the grants working group, as well as the board, considers whether such projects represent a vital research opportunity. And I'll tell you a little bit more about that. One of the other requirements that was set by the board was that the proposals that use convalescent plasma must also have a plan within the application for outreach and study participation by underserved and disproportionately affected populations, and that we would give priority to projects with the highest quality plans in that regard. The board also allowed the use of emergency INDs as part of the proposals. Next slide, please. So with that as background, we received a total of 19 applications that were submitted. Ultimately, 12 were eligible and were reviewed by the grants working group. And so of those, there were 3 that scored between 85 and 100 and thus were recommended by the grants working group. And the total amount requested from those 3 applications is just over $1 million. And we have 5 million available for those. On the next slide is just a brief outline of the 3 proposals that were recommended. There is one clinical study, CLIN2 project related to convalescent plasma. And there are 2 discovery projects that are recommended. And Mr. Sheehy, I can go through each of these, or I can go through all of them and then have discussion, however you'd like me to proceed.
Maybe if you could go briefly through these.
Okay, sure.
Because we have a lot to do. So maybe take.
Okay. All right, so so the first one is application 11775, and so this is the CLIN2. It's a clinical observation study that is related to convalescent plasma, and the goals of this project are threefold. First is to develop a screening program in California to identify potential COVID convalescent plasma donors. Secondly, to characterize the titer and neutralizing properties of anti-SARS-CoV-2 antibodies within the plasma. And then finally, to conduct a prospective analysis in the plasma recipients based on the clinical course of the disease and the convalescent plasma immunogenic profile. And so this application, as mentioned, is one that requires a vital research opportunity vote by the grants working group. So the vote, in order to pass, requires a 2/3 majority by the GWG. So this passed with 15 members voting yes, 4 members voting no, and 1 abstention. The application received a score of 85, with 11 scientific members scoring in the fundable range and 4 below it. The application in general was thought to be a strong application with a great team. There were some concerns that were brought up related to the overall program and the clinical trial outcomes, things such as details of the power calculation, the dose and volume that is required to monitor as part of the program to ensure that a correlation between the the antibody neutralizing ability and the plasma itself can be made. So there were several suggestions related to that that are outlined in the summary. And I also want to mention one of the other things that was not present in the comments specifically, but that was mentioned by some reviewers related to the plan for outreach to underserved communities. So although there was mention of it, it was not expanded on in the application. I think there was a desire to have that more fully explained by the applicant. So that's a summary of that proposal. I can stop for questions on this one before I go on to the next one. Okay, so the next proposal is a discovery, which is 11817. So this is a proposal that plans to use a stem cell-based platform to both identify T-cell epitopes for the SARS-CoV-2 virus and also identify T-cell receptors that recognize those sequences. And so ultimately what this project hopes to accomplish is to identify those epitopes that can be used for vaccine development. So it has the potential to help in that, as well as identify T-cell receptors that can be used for adoptive T-cell therapies. So it has the potential both for developing a vaccine as well as candidates for T-cell therapies. It also would develop the assay and validate the assay for additional screening of different proteins within the SARS-CoV-2 virus. So this proposal received the highest score of 95, with all members voting within the fundable range. This application had very little in terms of weaknesses, and no comments were made in terms of things that should necessarily be improved. I think reviewers felt that this was a very strong application. Okay, and then the last one is application 11764. And this is a project that would conduct a high throughput screen using lung stem cell organoids for compounds that would be able to either stop, well, both stop proliferation of the virus in the lung organoid model, as well as reduce the cell death of the lung cells. And so this project received a score of 85. There were 11 members that scored in the fundable range and 4 that scored below that. This project, again, was also, I think, very well received, had many strengths to it. There were 2 concerns. One was that it was unclear that the organoid can be infected with the virus or even inhibit infection. So it hasn't been, this model hasn't been validated yet with the virus. So there was a concern that that might be a problem. And some limitations in the screening. So the screening that they're doing is on about 2,000 compounds that are already FDA approved and may have expired patents. So there's an advantage to doing that, but it may also be limiting. So there was some suggestion that the screening either could be expanded or a rationale for which drugs are used in the screen might be reconsidered. So that's the final project. Mr. Sheehy.
Thank you, Dr. Sembrano. Unless there's any questions, I think I would proceed to a motion to not recommend for funding all of the applications that scored outside of the fundability range. So if someone would like to make that motion.
So moved.
Do I have a second?
Second.
So it's motion by Senator Torres, and then seconded by David. Could we get, is there any board comment on the motion or question? Could we get public comment? If there is any? So I see two hands raised. So maybe we could take the 310-825 phone first.
Hi.
And please state your name.
Hi, I'm Dr. Armo Swami from UCLA.
Okay. Please proceed.
Thank you
have
for
3
that.
minutes.
Thank you for the opportunity to comment on our TRAN application 11793, COVID-19 antiviral therapy to block lung stem cell injury and associated tissue damage. We need to urgently develop a therapy for COVID-19 as it is continuing to infect people globally and not going away. After infection, the SARS coronavirus 2 takes over the cell's machinery and establishes active infection, leading to cell injury and observed fatal outcome. Our screen identified a potent anti-COVID-19 drug candidate, belsosertib, which is already in several phase 2 clinical trials with a low side effect profile, which could be easily repurposed. We found that SARS-CoV-2 turns on a DNA repair pathway in infected human lung epithelial culture, and our drug belsosertib is specific inhibitor of this pathway, resulting in shutting down of virus replication and rescuing the cells from viral injury. Our score was lowered because reviewers asked for animal models, but we don't even know if animal infection replicates the human disease. And the primary human lung stem cell organoid model proposed is much more like patient lung tissue and a better study of efficacy. Recently, both empirical and experimental drug candidates in clinical trials have not shown expected benefit. Thus, we need to actively find new classes of drug candidates. So we request the committee to support this critical and timely COVID-19 drug study, which can help saving thousands of lives again. Thanks again.
Thank you. And then I also have a hand raised for a number beginning 650-079. If that individual would like to.
Do you see that one, Doug? I think Doug has to unmute the person. I'm not sure.
Yeah, I think the person needs to be unmuted.
Yeah, the 796-4063. Is that right?
Yeah, it starts with 650.
Mm-hmm.
Hello.
Hello.
Yeah, please proceed. You have 3 minutes.
Oh, yes, great. Thank you.
And we can get
I'm Albert Wong. Sorry. Yes, hi, I'm Albert Wong. I'm at Stanford University. I'm the PI of the rational design for a SARS-CoV-2 vaccine, number 11724. And as Dr. Zambrano mentioned, deliverables, our deliverable is a viable vaccine candidate. I just want to start by saying that of all the research activities we have going on, developing a vaccine is probably the most urgent issue. All the health and economic problems would simply go away if we had an effective vaccine against SARS-CoV-2. And our background in this area is that we developed a vaccine to treat glioblastoma, generally supported by a Tran Wen Award from CERM. I'm really happy to say that we would not have gotten as far and as quickly as we have without that support. And we, along the way, made 3 major advances in designing vaccines. Now, when we saw the press release in the request for proposals that came out, it was mentioned all hands on deck. You know, all scientists and clinicians should come together to try to fight the pandemic. And so despite not working on viruses per se, we felt really compelled to work on a COVID-19 vaccine because our insights from glioblastoma vaccine. Now, the problem is pretty much most of the groups are not going to have a lot of preliminary data on COVID-19 because it is so new. fact that the report that we needed to have the data to move forward didn't come out until the end of January. Our reviews frequently mention that we don't have enough preliminary data on the virus. We actually have an enormous amount of preliminary data supporting our glioblastoma vaccine, but just a small amount of that viral data that we're working to expand now. I would say it's much harder to make an anti-cancer vaccine than an antiviral vaccine, because basically we're trying to take a normal protein and make it look foreign. So that's why I'm optimistic about our vaccine development, because we believe we can make the virus, which is already foreign, look even more immunogenic. Now, it's not unusual for review committees to ask for more data, and we are going to reapply with our new data. But in closing, what I'd like to mention, the ICOC, is that some of the most interesting work that is going to be encountered is not going to have a lot of SARS-CoV-2 or COVID-19 data, and perhaps a path forward on to the GWG that they should take this into account when reviewing applications. So I just like to mention that I really do think that we could do something very important here. I hope that we can break away from this norm of constantly asking applicants to reapply with better data, because these are extraordinary times, and we have to have a different mindset if we're going to solve this virus problem very quickly. Thank you.
Thank you. We also have a public comment. I think it also begins 650. Gil, have you identified that one? I mean, Doug, I'm sorry.
I think it's the 626 number.
626 number.
Oh, I apologize. 626.
893-5033, and they are off mute, so they can speak.
Oh, that's why I can recognize them.
Mm-hmm.
Please.
This is John Zaya from City of Hope. Can you hear me?
Yes.
So I want to thank the committee for considering this. There were two major questions that were raised by the reviewers. Remember, this is an attempt to bring let's call it a service to the physicians in California who are seeking to treat patients with convalescent plasma. And we are linking a superstructure, which is the national program that links blood collection sites as well as the FDA to an infrastructure which is based primarily through the alpha stem cell clinics to reach out into the underserved areas. So one question was, how do we get to the underserved areas? And we do have already, let's call them fingers out into the areas through the alpha stem cell clinic. And I refer to that in a letter that I sent that has been posted, namely through the UCI Alpha Clinic. We have connections with the Riverside Native American community. And we will be working through that type of system throughout the state of California, using the alpha stem cell clinics as the way to get to the various Indian health services. The second thing that was raised is, what about statistics? Are you sure you're going to be able to learn anything from this? And we actually did describe the statistics in the clinical trial protocol, and I have reviewed that again with our statisticians. If there are 250 treated patients, then we will have significance if there is even 1 day difference between a patient who got treated with high antibody versus those who got treated with low antibody, or with some other characteristics of the antibody that we are going to characterize. For example, neutralizing antibody. So we're convinced that the statistics will be sound. Of course, every trial depends upon good enrollment and good collection of data, and we have that. We believe that we have that in effect. For example, already there are 154 physicians in California who have registered with the national study, and if each one of those treats only 2 patients, we would have way more number of patients than we need. So with that, I think I'll end, and I thank you again for considering our protocol, which is number 11775. Thank you.
Thank you, Dr. Zaya. And just a point, your application is not the subject of this motion, but it will be the subject of a following motion. So
Okay.
I
But
appreciate
you called my phone number, so that's why I spoke.
Oh, that's okay. No problem, John. I appreciate it.
Okay.
So do we have any other public comment for the motion? So if not, then the motion is not to fund any of the applications that failed to score within the fundable range. Could we call the roll for that, please?
Excuse me.
Yes, Ms. Durant.
Isabel Duran here. Just for clarification, for my purposes, being the newbie here,
Sure.
is there an opportunity for some of these? I think it was Dr. Wong who said that they were just getting more data that might create a more robust application. Can they reapply?
Yes.
Yes.
Okay, so we don't want to really eliminate the opportunities for people who have some really perhaps exciting, though less data driven kind of an application, because some of these things are like taking a flyer, but there's such a need for, I don't want to have them lose the opportunity to come back and perhaps to bring us some very exciting new ways in which they can do their research. Sometimes if we focus too much on hard science, we sometimes don't get the opportunity to be more perhaps imaginative and also to take more chances. And I think in this point in time, we need to take a little bit of a bigger chance. So thank you very much. I appreciate the opportunity to make my comment.
Yes, if I may respond to Isabel Artois here. Both JT and I made those comments before the grants working group where these grants were reviewed. And I will make them again today. We're going to have another review in mid-May of other applications that have come in, and we still have money left. So anyone is welcome to reapply or to apply newly for this second review that we're going to proceed with. And I'm sure Dr. Sembrano will give us the update as we move forward. Thank you.
I'd just like to add to that what Senator Torres just said. We have received the next round of applications already and are likely to receive further down the road as we continue along in these emergency sessions. So there definitely will be opportunity.
Thank you. Could we now proceed to the roll call?
Yes. Anne-Marie Dulege,
Yes.
Isabel Duran,
Yes.
David Higgins,
Yes.
Steve Julesgaard,
Yes.
Dave Martin. Dave, thumbs up. Okay, thank you. Lauren Miller,
Yes.
Adriana Padilla,
Yes.
Joe Panetta. Joe, you're on mute.
Oh, sorry.
Thank you. Francisco Prieto,
Am I unmuted?
you're unmuted now.
All right, yes.
Francisco Prieto, Robert Quint. Dr. Quint?
Sorry, I'm disconnected.
Oh, no, you're here. I hear you.
Yes, sorry.
Al Rolette? Al Rolette, are you still on somewhere?
Yes.
Oh,
Yes.
thank you. Jeff Sheehy?
Yes.
Stewart? Oh,
Yes.
sorry. Jonathan Thomas?
Yes.
Art Torres?
Aye.
Motion carries.
Thank you. Now we can either have a motion to approve all three of the applications in the funding in the fundable range, or we can take them one by one, whatever the desire of a motion maker is.
I move to approve all three.
This is Al Willett, I second.
Wait a second. Is there any discussion for the board?
Isabel here?
Yes, please.
I don't see the place where I raise my hand, so
Oh, I'm sorry.
I'm calling out.
Please go be my guest.
Thank you very much. I appreciate the follow-up letter we got from City of Hope to describe how it plans to reach out to make sure that underserved populations are being included. I'm wondering if there is an opportunity while they're doing it, or if they're giving us some midterm report to let us know how that is going, so that in fact they are meeting, in fact, their guarantee of inclusion. Can we have something to that respect? Some kind of midterm report about how they're doing in that, in terms of making sure that there is inclusion?
Perhaps
Proportional?
we could bring Dr. Zaya back on the line if he's still around. John, Dr. Zaya, are you still here?
Yes.
Yes, I would propose to make that a milestone.
So, and
Good for you,
maybe Mr. Ron, the way in which we typically do our grants, they get no go milestones at various points. So these are serious requirements that applicants have to make. It's part of the plan, and really it is tied to the ability to access further funding in their project. I don't know if that fits along the line. Is that a reasonable solution?
It sounds so. Sometimes verbiage is used that I'm not necessarily familiar with, particularly in the context of grants and proposals. And so I like the word milestone, and I'm glad that we're going to be able to, that City of Hope will measure that. I'm very pleased about that. But I'd love to see it in every single project in terms of proportional representation of underserved communities, no matter who's doing the project. But thank you very much, Dr. Zaya, and I wish you good luck on this.
Yeah, and maybe we could have someone from grants management interface with you and with Dr. Zaya. Like I said, they're very onerous. So the way in which we've evolved in managing our grants is to have very specific measurable milestones at points during the course of the grant. And those milestones are literal funding milestones, and you don't continue to receive funding if those milestones are not met. So
Right.
They're not soft, but we can, I think, I mean, Dr. Milan or Dr. Sembrano or Ms. Bonneville, if any of you want to kind of make a comment to that.
No, I think that's right, Jeff, and we can actually kind of meet with you, Isabel, and kind of give you an understanding of the milestones and also this program. And we hope to be able to give updates to the board along the way as these programs progress as well. So I think that there will be many opportunities, and the board members are always free to ask the CERN team for updates as well as all of the updates we provide in our public reports. Thank you.
Thank you.
Isabel again, thank you, Maria. I really appreciate that, so that I can understand, but I think it's as crucially important to our applicants so that they recognize that we are really interested, that it is very important to us to make sure that there is proportional representation in research of all of our communities in California, and of course our underrepresented racial and ethnic minorities. So I'm less concerned about my educational growth, but just want to know that these milestones are in there for applicants. So, and maybe they're already there in very strong language, but it's just something that I think is really crucial. So thank you for sounding like a broken record. Thank you.
No, please, I appreciate it. Thank you. Are there any other questions from members of the board? Or comments? Are there any, is there any public comment on this motion?
Seeing none.
I don't see any hands raised.
Okay, Maria, would you like to call the roll then?
Sure. Anne-Marie Dulege,
Yes.
Isabel Duran,
Yes.
David Higgins.
Yes.
Jules Guard,
Yes.
Dave Martin. Dave, thumbs up. Thank you. Lauren Miller,
Yes.
Adriana Padilla, Joe Panetta,
Yes.
Robert Quint,
Yes.
Al Roulette,
Yes.
Jeff Sheehy,
Yes.
Stewart.
Yes. Except for those with which I'm in conflict.
Why, thank you. Jonathan Thomas,
Yes.
Art Torres.
Aye.
Motion carries. Thank you.
Thank you, Ms. Bonneville. Thank you to the committee. And I want to thank the CERN team and our reviewers for moving on this so rapidly. And I want to thank Dr. Zai and the team at City of Hope. I think this is a therapy that we have potential to getting into patients incredibly rapidly, like all of the therapies people are talking about right now. We don't know if they'll work or not, but this is one that continues to show at least some viable promise. I will note, we will not be accepting applications for injection of Lysol or Clorox, notwithstanding our fearless leader. They're not in scope, but anyway, I will turn it back to Chairman Thomas.
Thank you very much, Mr. Sheehy. Just want to congratulate, echo what Mr. Sheehy said, congratulate all members of the CERM team, board and team for getting this first round of funding and allowing CERM and the state of California to play their role in this worldwide effort to come up with treatments for this disease. So we will be back to you with more on other projects at subsequent meetings, but I wanted to make sure that everybody, as we all do, understand the gravity and significance of this board meeting in connection with this condition. So thank you, everybody, very much. We're now at the part of the agenda for public comment on any topic of your choosing. Do we have any members of the public who would like to speak on any topic? Hearing none, then I just want to, in addition to Dr. Sombrano and Maria for all your help in pulling this all together. Thank you, Tricia and Doug, for all of your help. This is not an easy thing to pull together in short order, and everybody worked as a great team to make it happen. So thank you all. And with that, we stand adjourned.
For those of you who on the application review committee, if you could fill out the poll for the week of May 11th, if you haven't already, so that we can schedule our next meeting, that would be lovely. So thank you, everyone.
Thank you. Bye-bye.